She will do a general introduction, then Dr. Saad will hopefully say nice things about you, and then we'll bring the lights down.
Thank you very much.
Good morning, everybody. Welcome to our first grand rounds of 2020. And before Dr. Saad introduces our guest speaker, Will Zacharias, I want to say a few things about Dr. Saad. I've known him since I've been here since 2000. He joined in 1999. If you ask him what his most prestigious accomplishment is, you'll see how humble he is. He's happiest that for an entire year there were no Clabsy and Caudy in the ICU. He's worked diligently for infection control. He won a Beacon Award in the late 2000s for this. He's therefore was recognized and asked to cover the ICUs and take some of the same quality control in the ICUs at Jewish Hospital. And if we didn't have people like him, we wouldn't have a transplant program either. So I really want everyone to say thank you very, very, very much, Dr. Saad, and come on up and introduce your speaker.
Thank you, everyone. Good morning. It's actually a pleasure for me to introduce one of the speakers from the neighbor, of course, areas in the University of Cincinnati. We've been doing this program now for a few years, where we invite speakers from UK and Cincinnati. And our guest speaker today is outstanding scientist. He, all through his career, has been distinguished, did his college in Northwestern and moved to do his medical school in the University of Michigan in Ann Arbor, and then his residency at University of Pennsylvania, and he did also his fellowship there. And of course, you know that in the University of Pennsylvania, they have 4 years track, so he's a scientist, and he spent an extra year after his fourth year as a postdoctoral fellowship there, and then started his academic career at the University of Cincinnati in 2018. If you look at his CV and his accomplishment with the Alpha Omega Alpha when he was in medical school and Dean List for research and several awards, tell you exactly where his career is going to be. I foresee that probably in a few years he's going to have his R01 and he's going to be well known. The area that he's going to be talking about is actually a very exciting area because we deal with these patients in the unit with the acute lung injury and ARDS, and his area of expertise is looking at this mechanistic area of regeneration and healing and who does well and who doesn't do well and how we can predict that. And he's going to tell us a little bit about his work today, so I'm very excited to hear about that. I want to thank him for coming, and thank you, Dr. Zacharias.
Thank you.
All right, well, thank you, Dr. Saad, for the kind invitation. It's a great honor for me to be able to be here, particularly for the first grand rounds of the year. My academic interest, as Dr. Saad mentioned, is in how lungs regenerate after severe injury. And what we have focused on is trying to understand exactly how the different cells activate and what the mechanisms are that could allow us to develop some therapeutics. And this really started for me actually when I was a junior resident. I admitted this patient to the medicine floor, and because I had pissed off my chief residents, I then admitted her to the ICU the next day because I was on call 2 days in a row. So this was a 38-year-old woman who had mild asthma, smoked, knew she shouldn't, had a kid who was in second grade who came home with a viral illness, hadn't gotten a flu shot, and ultimately ended up with influenza A. I admitted her to the floor where she started off on about 2 liters of oxygen, looked okay, no major issues. And unfortunately, the next day when I was on short call in the medical intensive care unit, I intubated her, and within about 6 hours before I went home that day, crashed her onto ECMO for severe ARDS. So she had an incredibly precipitous presentation, ended up on ECMO for 6 days. And after she was on ECMO for 6 days, they were able to wean it. I admitted her again back to the medical intensive care unit where we weaned her off the ventilator. And occasionally now she sends me pictures of herself and her kids out around the country doing various exciting things. Normal, basically uncomplicated post-ICU course. And she followed with me as a primary care patient and then with me as a fellow for her mild asthma, for which I did basically nothing. And so I was struck by this course and thought, this is incredibly severe disease. It started really fast, it ended really fast, and this is a great outcome that we all hope for with all of our cases. Why did this lady get better, and how can we make other people get better in the same sort of way? And when you start to think about the complexity of the clinical problem that we're talking about, what you realize is that it's remarkable. I have this slide in mostly for the scientific presentations that I give, but I think it's probably intuitive to everybody in this audience that we're very used to dealing with incredibly severe infiltrative lung diseases from acute illnesses. So when you see someone with ARDS, you see a profoundly abnormal chest imaging study, and then you sort of expect that if you see these people 6 months later, they're going to have a normal CAT scan again. And that's actually totally remarkable when you think about it. And it gets even more complicated when you realize that the histological injury that you're talking about underlying that radiographic finding is really profound. So this is a mouse that received H1N1 influenza. This is actually the 2010 strain of H1N1 influenza. It's been adapted to mice. For the research aficionados in the room, this is the PR8 strain of influenza. And what has happened is this mouse has developed incredibly severe lung injury with areas of almost total obliteration of the alveolar structure, opposed to areas that are relatively normal looking. And we know this is true in all of the lung injury models that we see clinically. There's a tremendous amount of heterogeneity, and what I'd like to try to convince you today is that the mechanisms by which a mildly injured area and a severely injured area regenerate are going to have to be different, and that we need to understand what each of those two mechanisms is in order to be able to develop an effective therapeutic for lung regeneration. So what I'd like to talk to you about today a little bit is just give everybody some background on lung regeneration and what sort of the cells are that are involved in the process of lung regeneration. I'm going to tell you a little bit about the progenitor lineage that we study in my lab that I was fortunate to be able to initially describe during my postdoctoral research at Penn. And then I'd like to end just talking a little bit about how I think that physician scientists are important in developing strategic clinical trials to try to translate some of this knowledge to help our patients. And I'm going to warn the residents, you're going to think more about what happens inside a cell in the next hour than you have since M1 year. Sorry about that. Okay, so this is my schematic diagram of lung epithelium. I think it's everybody knows that there is an airway and there is an alveolus. This is an oversimplified diagram, but there are a number of controversial lineages in the lung that do out different things. So the basal cell, which sits at the base of the epithelium of the airway, is a professional stem cell. And it makes secretory cells and ciliated cells in the epithelium of the airway. Under the right circumstances, cells in the smooth muscle gland can take on some of that as well, and neuroepithelial bodies can also generate some airway cells. I'm going to talk about a small group of stem cells that come from the airway and involve the alveolus in a second, but that's about it that I'm going to say about the airway today. So we're going to focus mostly on the alveolar portion of the lung, which is lined by a combination of type 1 epithelial cells, which sit in close approximation to the alveolar capillaries, and type 2 epithelial cells, which coordinate a variety of immune functions, make surfactant proteins to try to maintain surface tension, and also are facultative progenitor lineage in the lung. So let's start by talking about basal cells that come out into the lung. And this is important because this is a common phenomenon in influenza-mediated injury. So this is another different version of a mouse with the same influenza virus. And what they've done is they've given the mouse flu, they've waited 15 days, and this is a cross-section of all the lobes of a mouse lung looking for staining for two things. The green stain is a type 1 cell stain that shows normal-looking alveoli. The red stain is for keratin 5, which is a marker of airway basal cells and should not be out in the alveolus. And what you see is that there are these cords of keratin 5 positive cells sort of juxtaposed to areas of relatively normal-looking injury. This highlights the point I made earlier that there are areas in this mouse that are very uninjured, but if you look at the whole amount of the lung, you can see it's almost moth-eaten There's so many different areas of injury. And this animal died at day 15 and would have died whether they sacrificed it or not. This is a severe acute lung injury that leads to the death of the animal. And what you're seeing is that there's a number of areas that are relatively normal. What they don't highlight in this paper, and I'm going to come back to later, is these areas that are blue. So blue is a cellular stain called Dept. It just shows the nuclei of all the cells. And there are epithelial cells in these regions that are blue. But they don't talk about what happens here, and I will as we go through the rest of this talk. Well, let's focus on the keratin 5 positive regions for a second. These areas, when you look at them, are disorganized sort of messes of epithelial cells. They develop within days after the initiation of an influenza injury, and they persist, as far as we can tell, for the life of the animal. And they develop into cystic-like structures that look very much like the cysts that we see in lung diseases like scleroderma-associated fibrosis. And these are important lineages in that they try to become functional alveoli. They start to develop lumens, but they have abnormal signaling sort of throughout their entire cell surface, activation of a notch signaling pathway that's abnormal. And these make cysts and cords. And you may say, well, okay, that's a mouse. What happens in a human? This paper is from a 31-year-old gentleman who developed severe H1N1 influenza in Boston, went to a famous hospital there, was on ECMO for 9 months, and ultimately received a lung transplantation because of failure to wean from ECMO. And what you can see in the explanted lung is these huge cords of cells that are thick. These don't look anything like the alveoli I showed you earlier. And what they are is they stain for this basal cell marker, keratin 5, suggesting that these severe, acute cords of cells persist in humans as well as in mice. So what is this for? Why are these here? Well, the reason that they're here is because holes in the lung are really bad for you. So this should be obvious to everybody in this audience. It's not always obvious to my scientific colleagues. But what happens after you develop a severe acute injury, if you pop a hole in your lung, is a tension pneumothorax. And before the invention of the chest tube, which I'll remind you in geological time was relatively recently, you would just die if you had a tension pneumothorax. So there's an evolutionary pressure against having a hole in your lung that should be sort of intuitive. And what these cells do is they come out very quickly and they fill the hole. They don't really care if they're making a scar because the goal is not to fix the lung, it's to prevent the death of the organism. A number of labs now are working on trying to take advantage of this phenomenon that once the cells are there to try to convince them to do the thing that we would like them to do, which is then regenerate this region into functional lung. But that's not really their job. Their job is really to keep the animal from dying long enough for regeneration to be relevant rather than simply death of the animal. And it turns out that this is intrinsically makes sense at a molecular mechanistic standpoint. These cells are driven to the parts of the lung that are most hypoxemic. So they're activated by a pathway called HIF1 alpha, which is a downstream transcriptional regulator of hypoxemia signaling. It should be obvious to the clinicians in the group that the areas with the most severe lung injury are probably going to be the most hypoxemic regions of the lung because they're not getting good gas exchange. These are the areas that we generally think of as shunting away from with our hypoxemic vasoconstriction, or if we don't, getting shunt physiology that then requires us to ventilate the patient to make sure that they maintain their tissue oxygenation. And those hypoxemic areas then drive in these keratin 5 positive cells, and the absence of HIF1 alpha signaling obliterates this phenotype and changes the phenotype of the cells once they get there. The second cell I'm going to tell you about is a cell that, as far as we know, is in the mouse only, but we think that from some work from my mentor's lab, there may be a similar cell in human that's going to be described soon. And this cell is called the bronchoalveolar stem cell. It's a cell that sits right at the junction of the airway and the alveolus in the mouse. And these cells were originally described in 2005, and it took until 2019 for someone to prove that they really exist. So in 2005, they were shown by immunostaining to be a combination of airway and alveolar markers at the bronchoalveolar duct junction, and they were proposed to be a progenitor cell. Someone had to do a very complicated double lineage tracing study to actually demonstrate them. And what this group did in a beautiful paper that came out last year is they used an SPC Cre, which marks type 2 cells, and a CC10 Cre, which marks secretory cells in the airway. And they used a lineage reporter that required both of these two alleles to be expressed to make a cell red. And what they showed is that these cells exist at the bronchoalveolar duct junction. If you injure the airway, they tend to give rise to airway lineages. If you injure the distal part of the lung, they tend to give rise to alveolar lineage. But there's less than 1,000 of them in a mouse lung. They're very, very rare. And what this cell is a cell that is your backup system. If you lose all your professional progenitor cells in either the alveolus or the airway, these cells get activated. You don't need a ton of them because they're really your emergency backup. But again, holes in the lung are super bad for you. So having a useful cell that can back up other lineages if you start to lose their capacity and regenerate functional portions of the lung makes a lot of evolutionary sense. And so that's what this cell does. I'm going to spend the rest of the next probably 30 minutes of this talk talking about the alveolar type 2 cell. And the alveolar type 2 cell is really a very important cell in a number of different contexts. But what I'm going to tell you about today is its progenitor function. So if you take a mouse or human lung and you grind it up into a single cell suspension and you pull out an individual type 2 cell and you mix that type 2 cell with about 100 fibroblasts from a lung and you put them in a three-dimensional culture, they will grow something that looks like this. And this is called a lung organoid. Those of you who are interested in science may know that organoids were named the technology of the year in 2019 in Science magazine. But what they are really is individual small three-dimensional culture systems that recapitulate some important aspects of the organ under study. You can grow them from induced pluripotent stem cells. You can grow them from primary cells. In the lung, the induced pluripotent stem cell technology is not sufficient yet to be able to grow a fully functional organoid from iPS cells, so we grow them from primary cells. So what you see here is a clonal organoid. Every epithelial cell in this organoid has developed from a single type 2 cell. What you see is purple cells here that are type 2 cells and express the type 2 cell protein SPC. And then type 1 cells, which line the inside of this lumen, which express the type 1 cell protein HOPX. And this is and the other cells that aren't labeled with either of those are fibroblasts that have intercalated into this ball of cells to try to generate something that looks like a functional organoid. And the inside of this has a lumen. The lumen's functional. They secrete things in here like you think they should. If you stuck a needle in this and pulled out the protein, it would have surfactant protein in it because they're type 2 cells here. So this is in a lot of ways a functional organoid. The reason this experiment was done was not actually to generate something that we could study, though. It was as a stem cell assay. So you can imagine that most of the cells in your body, if you take them and stick them in a three-dimensional culture, will not make an organoid. Only a very few number of cells, progenitor cells, in various organs will make organoids under these conditions. And so you can use this assay to determine the potency of a cell as a progenitor cell. And so that's how this was done, and this was done in a beautiful paper by Bridget Hogan at North Carolina a few years ago. So we decided to go in and try to see if we could find a subpopulation of these type 2 cells that were enriched for this progenitor capacity. Because there's millions of type 2 cells in your lung, and it's hard to believe you're maintaining millions of type 2 cells that are surfactant factories doing a good job of maintaining your immune homeostasis in your lung and functioning as progenitor cells. So we had the hypothesis that a subselection of these cells would be the cells that are doing the regeneration and that we could figure out what they were using mouse genetic tools. So what I'm going to talk about for the next 10 minutes is how we figured out which subgroup of these cells is type 2 cells, is the type 2 cell progenitor. So we turned to a complicated mouse experiment that I'm going to explain. Okay, this is called lineage tracing. And what you do is you take a Cree, which is activated by Wnt signaling, in this case with the promoter axon 2, and you combine that Cree allele with a second allele that's in a locus in the mouse called the Rosa locus, which is just a safe locus to stick some genetic material. When these two alleles are active in the same cell, what happens is the cell turns green. And then every single cell that comes from that cell will be green for the lifetime of the animal. So you can mark the cell itself and all the cells afterwards. In addition, our Cree has a second fluorescent protein in it, which is red. So it allows us to separate cells into the lung into three categories. Cells that are red but not green, those cells are responding to Wnt signaling right now. Cells that are green but not red, those are cells that are came from a cell that was responding to Wnt signaling at the time you marked the cells, and cells that are both. Those are cells that are from the lineage and responding to Wnt signaling right now. Okay? And I'm going to talk about red, green, and yellow cells going forward. And so basically, the first question we asked is, where are the green cells? Where are the cells that are marked by Wnt signaling, and what does that tell us about what Wnt signaling is doing in the lung? And it turns out they're a 20% subfraction of the type 2 cells. So we've now identified that 20% of the type 2 cells have this Wnt signaling signature, and this was a good guess that this was going to be the progenitor lineage because Wnt signaling is involved in stem cell biology in a number of organs, particularly those derived from the endoderm, like the stomach and the intestine and the liver and others. So we went on to ask what happened to these cells over the course of the lifetime of a mouse. So the way I did this experiment is at 8 weeks of age in the mice, I gave them a series of drug injections with tamoxifen to activate the Cre, and then I waited for 9 months. And this is why my fellowship took 5 years, was because I was doing experiments where I had to wait for 9 months. And what we saw basically is that over the course of the lifetime of an animal, the Wnt lineage started to contain a slightly increased number of alveolar type 2 cells and a significantly increased number of alveolar type 1 cells, consistent with the homeostatic turnover of the lung. So about the average lifespan of an alveolar epithelial cell in a mouse is about a year. It's a little bit longer in humans. So the turnover is relatively slow. So if you do the kinetic analysis of this, what you see is that the number of type 1 cells that are made from this lineage are basically the replacement rate of type 1 cells over the course of the life of the animal. So when we saw this number, we thought, oh, hey, that's interesting. It gives us the opportunity to think to ourselves about what this cell is doing that's different if it's doing most of the homeostatic turnover in the lung. And then we started to ask whether these cells were from the lineage or whether they were still responding to Wnt signaling, and also whether any new cells came into this lineage over time. And to our significant surprise, it turns out that every cell that's green, up to 9 months, with very minimal exceptions, is also red. So virtually all of these cells came from the same lineage, and there's essentially no drift of other type 2 cells into this axon 2 positive lineage, into this Wnt responsive lineage over the course of the life of the animal, which was a big surprise to us. And it suggested that this type 2 cell population that we'd identified was actually pretty stable over the life of the animal, which gave us some hint that it might be important in a regenerative process. I'm going to skip that, but at about the same time our paper came out, another group from Stanford showed about the same thing using a different axon 2 Cre, also showing a relatively similar homeostatic turnover. So the first thing I did is I pulled these cells out by fluorescence-activated cell sorting, and I put them into the organoid system to see if they could make organoids. And the answer is they do. They make organoids. They make bigger organoids. They make more organoids compared to the whole type 2 cell population, suggesting that this fraction was enriched for the progenitor activity that we were interested in. So following that, we decided to go back to the flu model and try to figure this out. And the reason I picked flu is because there are a remarkable number of bad lung injury models that people use in the literature, and I suspect this is true for most of your organs as well, where people will say, I did X experiment, and that's a model for pulmonary fibrosis, blah, 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 and most of them are not good clinical models. And actually, one of the jobs of a physician scientist is to say what is a good and what is not a good clinical model for a disease that we actually see. What I like about the flu is that it has 3 characteristics that I think make a good clinical model. The first one is it's the same viral injury. So it's the same injury as what we see in humans. The same virus, it's the same subtype of virus that caused a pandemic outbreak in humans. So that's probably tells us that this is a virus that matters to our biology, and if it matters to mouse biology too, then we can use it as a model. The second thing is that it causes pathology that you expect in a human lung when injured with the same virus. So what we saw when we injured these animals is essentially 4 histological states. We saw areas of normal lung, we saw areas of what a human pathologists would call hyaline membranes disease. You guys see the pink stuff on the edge of these alveoli? That's surfactant protein that fell out of dead type 2 cells that's got slammed up against the side of the alveolus. Turns out that's super bad for gas exchange because all that surfactant protein blocks the area where oxygen molecules are supposed to go. And so that's why people with these sort of injuries get hypoxemic, even if their CAT scan doesn't look that bad. It's because they've got hyaline membranes disease you can't see at the CAT scan, and that's causing them to have altered gas exchange. This pathology is what a pathologist would call, on the spectrum of fibroproliferative ARDS, which is you're starting to see a fibrous and infiltrative sort of feature here. And this is thought of as a regenerative state in human lung, but in mouse it's just another injury state that we see. And then this is what we called alveolar destruction, where I think I don't have to convince you that that doesn't look much like a lung. So this is an area where the immune infiltrate is so profound that it's basically knocked out all of the functional alveoli. This is exactly what you see if you take a flu ARDS patient who dies and you look at their lungs with autopsy. So that's step 2. You've got a model that looks like it's a relatively real thing. And then the third key part of this is that it is a model that you can actually study. So you can generate these animals in relatively significant bulk, and the regenerative process takes a couple of weeks. So you can study these in about 4 weeks, which is relevant because you don't want something that takes 9 months to cause an injury in mice, because you don't have 9 months when your patient has flu ARDS. You have a couple weeks at best. And you want to be able to look at a similar time scale as what you have to humans. So for all those reasons, we picked this flu model and instituted it in the lab. And then we used it with the lineage tracing I described to you earlier to try to understand what the kinetics of these cells were after injury. And what it turns out is that in areas of minimal injury to the lung, essentially nothing happens. The cells sit there. There's 1 or 2 of them per alveolus. That's what we see in homeostatic conditions. And so nothing occurs there. When you start to see injury, you start to see things like this, where you see doublets of green labeled cells. This is expansion of the type 2 cell population. The majority of new type 2 cells are coming from this green lineage. And in areas of more severe injury, you start to see these cords of type 2 cells that start to expand. And you can see that this profound infiltrate here, many of these cells are actually positive for the green marker, suggesting that lots of these cells are coming from the lineage. And when we looked at this by fluorescence activated cell sorting, we saw sort of a remarkable phenomenon, which is we saw before the flu that the cells basically fell into 2 categories. They were either green or they were nothing. So they were green and red, or they were not labeled. After we give the flu, we see an evolution of a green-only population that gives rise to a significant number of new epithelial cells. And we see self-renewal in the progenitor cell population, but we see almost no recruitment of other cells into this progenitor cell fate, suggesting that your regenerative capacity after a flu injury is the cells you have before the injury. You can't recruit new ones during the injury, which is really important to know, right? Because that tells you which cells you have to target. If you can recruit new cells, what you'd like to do is take all the nearby cells and recruit them into the progenitor lineage. But if you can't recruit new cells, you've got to use the ones that are still there and make them work for you. And so what this data suggests is that there's a significant requirement for targeting this AEP population, the axin 2 positive type 2 cells, rather than trying to recruit other type 2 cells into that lineage. The way the cells do this regenerative activity is by entering the cell cycle preferentially. I'll spare you the basic science details, but essentially we did a number of proliferation markers, and what we see is that the vast majority of the proliferating type 2 cells come from the axin 2 lineage, suggesting these cells are primed to enter the cell cycle, and that's how they regenerate the lung. I told you earlier about that keratin 5 cell that fills the holes. Well, here they are in red. They fill up that zone of total alveolar destruction. So that's the area that's the most hypoxemic. That's where the cells go. And in the rim around that is where you see the most robust activation of this type 2 cell progenitor population, suggesting that those cells fill up the area that's unregenerable, needs to just be filled up and prevented from scarring. And then the regenerative function occurs right around that in areas that are not quite as hypoxemic, where those cells don't come in and invade. For the scientific aficionados in the room, we did the lineage trace in both directions to try to see if these populations were ever able to switch between each other. And the answer is they're not. They're completely exclusive lineages. And I never saw a keratin 5 positive cell that came from the axin 2 lineage, nor an axin 2 positive cell that came from the keratin 5 positive lineage. So then the real question is, does it make lung that we care about? And the answer is yep, but it's really slow. So this is 3 months after the injury in mice. And what we see is these keratin 5 positive areas are still here. I told you the border region was where we saw the most robust activation of these type 2 cells, progenitors. And what we see is that the vast majority of new epithelial cells in these newly formed alveoli at these border regions bear the lineage mark. So almost all of the regeneration that's happening right next to this really severely injured region are coming from this one cell population. So the second most injured part of the lung regenerates, and it regenerates using the activity of this axon 2 positive AT2 cell, which was really an exciting finding because it suggests that this is a key regenerative lineage in the development of effective ventilation after a severe lung injury. So now let's talk about humans. So we got these results, and I got pretty excited because I thought, wow, this is cool. This lineage does a lot of really good, interesting regenerative things. That was a hell of a lot of mouse work. Maybe we can figure out how to make this work in humans. And so I did an experiment that I think is a good example of an experiment that only a physician scientist would be dumb enough to do, which is I took a transcriptome data set from RNA-seq comparing the progenitor cells to the other type 2 cells, okay? So we were just looking for gene expression differences between the two lineages. And then I looked for cell surface markers that were expressed just on the stem cell, okay? So the ones that were just on the progenitor cell. And then I bought a whole bunch of antibodies, and I screened by fax to try to find a cell surface marker that would purify out the progenitor cell, with the idea being that if I could find a cell surface marker in mice that would purify out the population so I didn't have to use this complicated mouse genetics approach to find these cells, that maybe I could try that on human lung and see if I could pull the same fraction out of human lung. And it turns out, after a substantial number of experiments and some good luck, that we can actually do that. So we found this marker, TM4SF1, which is involved in protein kinase C signaling and is a very interesting protein. For cell biologists in the room, it works in lipid rafts, concatenating signaling complexes on the cell surface, and probably coordinates the complex that drives canonical protein kinase C signaling at the cell surface of type 2 cells. And I'm happy to talk more about that if people have questions later. But in this case, we used it purely as a marker. It marks 99% of the type 2 cell progenitors in mice. And so if you take a human lung, and the way we got the human lungs at Penn, how we get them at Cincinnati, how I imagine you guys get them, is from organ donors. So what we had was a pathway with our organ procurement organization. When the lungs were good, they went to lung transplant. When the lungs weren't that good, they went to something called ex vivo lung perfusion, which sometimes regenerated them for transplant. And when they weren't so great, I got them. And that was my normal. And so in order to try to pick an area of lung that was actually like closer to normal, considering that these people by definition had had a really bad day the day before they gave me their lungs, was to take the most healthy region of the lung we could find grossly. And it turns out actually that when you look at the histology of these regions, it's remarkably preserved with cold ischemia. So we were able to get actually really good quality lung. And we were able to get really good quality, viable cells that we could use for a variety of downstream analyses. So if you look at the human type 2 cell population, which is marked by the marker HT2280, the human TM4SF1 separates that population really cleanly into two populations, the type 2 cells and the presumptive progenitor lineage. And when we took that progenitor lineage and we did an RNA sequencing experiment to see what it expressed, the thing that it expressed at the highest level was the Wnt signaling pathway. That should make you happy. It made me very happy because it means that not only is the TM4SF1 expression the same, but probably the molecular signature is similar between the cells. And this may not seem like a lot, 36% conservation of transcriptome between human and mouse, but it's actually a massive number. Random cells tend to be like 5%. So 35% is massively enriched and suggested to us that these really might be functionally equivalent cells. So we turn to our organoid assay, and this is sort of the killer experiment of my life so far, which is that I could separate out the AT2 cell population, which made organoids, into two different populations, the progenitors and the other type 2 cells. And when you take the progenitors, they make massive organoids. And when you take the other type 2 cells, they sit there. They do nothing. So this tells you that in human lung, we can separate out the progenitor population of type 2 cells with two markers, the HT2280 and the TM4SF1. which means in principle, from any human lung sample, I can now pull out the progenitor lineage. So that means from your patients with bad injury, bronchoalveolar lavage samples, et cetera, we can in principle do these things. And so this was a really exciting experiment for us because this, to me, opens the door to start to think about how we can really start to translate some of these findings from mice in humans. And so the first thing we did is we just looked at the signaling pathways that were really enriched in common between the mouse and the human. And it turns out one of the key things that's enriched on these cells is a receptor called FGFR2, which is the canonical receptor for the ligands FGF7 and FGF10. For some of the more seasoned folks in the audience, this is KGF and KGF2. And the KGF is probably the best studied pro-regenerative ligand in lung biology. And what we found was that only the human AEP responded to FGF7 and 10 ligand, and it responded massively, causing the biggest organoids I've ever seen that just proliferate out of control and overgrow the dish. So we can use our ability to pull out these cells to identify potential pathways that we can use to manipulate their function. So what I'd like to leave you with in terms of the stuff that we've published is this model, which is that under normal circumstances, there is a sublineage of type 2 cells that we call the alveolar epithelial progenitor, that they're largely quiescent, that Wnt signaling promotes the stability of the progenitor lineage, and that after injury, these progenitors re-enter the cell cycle, robustly regenerate areas of alveolus, and they do so under the combination of Wnt and FGF signaling, which we think leads to a model of cell proliferation and alveolar regeneration after injury. And this diagram, trying to figure out the precise molecular details of these intracellular pathways, was the subject of my K08 award, and we're working on that right now. But let me tell you why I think this really matters, which is the other patient I admitted the same day. So this lady was 39, the other one was 38. This one didn't have asthma, did smoke a little bit too. She also didn't have a flu shot. She caught hers from her coworkers instead of her kids. She also came in. They put them in rooms next door to each other. You guys know when you admit patients and you can't remember which one's which because they're the same damn presentation? They almost look the same. And so I was trying to remember who was who because it was the same story. Well, the next morning I met them both again because I crashed her onto ECMO the next day too. And she didn't get better. And so this happened when I was like a 4th month junior resident. This one died. The other one got better and followed with me in clinic for 6 years. And to this day, it bothers me that we have no idea why this happens to people differentially and how we can start to think about the process of getting these people off of these really advanced support things that we're now very good at doing, right? It's super easy to put someone on ECMO. But your ability to control getting them off of ECMO or off of the ventilator is basically zero. And when your patients ask you, like, what do? You say, we wait and hope it gets better, which is incredibly unsatisfying, right? And that's the state of the art in acute lung injury. So you guys may think, well, ARDS, if they get better, they get all the way better because that's been the classical teaching. But it turns out only 80% of patients with severe ARDS who recover have normal lung function a year later, which means that 80% do better without you having to do anything, which is good. 20% do super badly without you having to do anything, and since you have nothing to do, that's bad. So you're stuck, and you literally just have to tell them, wait, and I hope it gets better. That's very unsatisfying. So what my lab is focused on now is trying to understand the mechanism of progenitor cells like the one I've described to you to try to generate enough mechanistic understanding to be able to actually develop a therapeutic to meet this unmet need. So I'm going to finish just by talking a little bit about what I think is the future of how we can take basic science like this and translate it into functional capacity to be able to actually help our patients in the ICU. So the first thing is we have to really understand the cell. And this may seem like a really sort of esoteric statement for folks who are working at the bedside. When I say, is this cell a state or a fate, meaning is this a defined thing that is at the end of a differentiation pathway, or is this something that any cell can become? But understanding whether a cell is in an immutable defined state versus whether it has plasticity is a key portion of understanding whether it's a reasonable therapeutic target, right? So if the cell's differentiated, it's fully, completely differentiated, then your ability to change the way that cell functions is limited by the activation energy of having to change a whole proteome in that cell, and so it's going to be a hard target. Whereas if a cell is in a state that's sort of more of a superposition before differentiation, you might actually be able to target that cell. So we've been doing a series of mathematical modeling studies to try to combine transcriptome and epigenomic data from these cells to try to identify pathway targets. I'm going to skip the details and just tell you this. We did a lot of math. MATLAB's better at it than me, to combine the epigenomic accessibility of the cells and the transcription factors the cells express to try to generate something called a gene regulatory network. A gene regulatory network is like a wiring diagram for a cell. It tells you about the nodes of transcriptional activation that can lead to effector pathways that you want. So from a clinical standpoint, what it tells you is what would you need to target to make a cell do the regenerative thing that you want it to do. So to understand that, we did a series of complicated epigenetic studies to try to define what the key transcription factors are, and this is the network that we came up with. And so the question when you come up with a diagram like this is, does this make any sense? Well, I told you already that the cells respond to Wnt signaling. What's in red up there are direct Wnt signaling targets, so that made sense. I told you they respond to FGF signaling. Those are the direct FGF signaling targets. And I told you TM4SF1 is a protein kinase C signaling pathway. And I guess I took that slide out, but there are protein kinase C signaling targets in here as well. And so what that tells you is that this meets sort of your criteria for reasonableness. You have an idea of what it is. And so what one of my graduate students is doing right now is trying to dissect each of these nodes with genetic experiments in mice to try to identify which are the key nodes in the regenerative activity of the cell. Because if you knew what the key transcription factors were, then I would propose to you that you might want to know what drugs target those transcriptional pathways and whether you could give them to a patient with ARDS. Because those are the sort of targets that might actually work for somebody. So the other thing that you need to understand when you're thinking about regeneration is what is the context in which that regeneration is happening, the other cells that live nearby. One of the reasons that lung biology is a little bit behind, say, cancer in our understanding of our diseases is because there are conservatively 45 different cell types in the human lung, and they're all very, very close to each other. So trying to understand the signaling interactions between these different lineages is a critically important part of understanding what's really wrong in our patients. And so I did some work with a friend of mine who is starting his own lab soon, I think at Penn, Jared Dept, who defined a niche fibroblast that sits right next to the progenitor cell that I told you about and has a number of really important relationships in interacting between those two cells. So maybe you can't change the fate of the epithelial cell. Maybe that cell's hard to target. But if you can target the niche and make the niche tell the epithelial cell what you want it to do, maybe that would be a pro-regenerative therapy. So understanding these targeting interactions around the cell that you're interested in give you some sense of what the right way to move forward could be. And actually, taking some of that work has allowed other labs to start to grow organoids from mouse lung without any fibroblasts. So the system I told you about, you had to mix fibroblasts with epithelial cells, so the epithelial cells would have a niche to grow. If you take all the good stuff that those fibroblasts make and spend $1,000 and put it in the media, you can grow the epithelial cells without the fibroblasts, which suggests relatively strongly that it's the factors from those fibroblasts are key to the function of the cell, and then you can sort of start to dissect which of those is most important and which ones are druggable. Okay, so then you say you've identified the cell you want to target and the pathway you want to target. Now you got to get something to the damn cell, and that's a real problem, particularly in a sick lung. So the people in this room who have more experience, particularly in critical care, will remember that at various times people probably stood up here and talked about activated protein C for sepsis and albuterol and steroids and all sorts of other therapies for ARDS. And guess what? All of those clinical trials failed. And the reason they failed, I'll tell you about in a minute, but part of the reason why they failed is because it's hard to get drugs to the business end of an ARDS lung. The cells that you actually need to target are in the areas that are sick. It's hard to get stuff there. There's fluid in the way. You've shunted the blood away from it because you're trying to avoid hypoxemia, And so it's a challenge. So how do you target AEPs? Well, one way that we started to think about doing it was with genome editing. And in a paper that we published in Science Translational Medicine last year, we used modified adenovirus to deliver gene correction for a lethal lung disease to mice. So the way we did this experiment is we took a mouse that had a severe surfactant protein mutation that was uniformly lethal. We designed a virus that used CRISPR-Cas9 genome editing, which you may have heard of on the news, unfortunately, because the news has no idea what CRISPR-Cas9 actually is. I want to point out these are baby mice, not humans. Don't get me in trouble, okay? I wasn't that guy. Someone else did that. But what we did is we took this virus, we designed it to knock out the deleterious allele. So these animals were heterozygous for a dominant active mutation that killed them. We designed a CRISPR construct to knock out that dominant active mutation. We put it in a viral envelope, and we injected it into the amniotic fluid of the pups at E16.5. It was a couple days before birth in mice. To remind you guys of embryology in medical school, babies breathe amniotic fluid. So they breathed it into their lungs. The virus went with. And what you can see here is that with no virus, there are no green cells in the lung. And with a virus that turns the cells green, there are a lot of green cells in the lung. So we were able to efficiently and directly target alveolar epithelial cells and only alveolar epithelial cells. This was sufficient to actually improve the mortality in this mice. 100% of the mice died without the genome editing, and 80% of the mice died with the genome editing. So we were able to save 1 in 5 mice simply by editing out this allele in a relatively short period of time prenatally. And so this suggests that if you had the right cellular targeting, you can derive real therapeutic benefit. We think this has significant applications for pediatric disease, and I'm at Cincinnati Children's Hospital for my lab, so we've been trying to follow up on some of this stuff. But I actually think this is also really exciting potentially for adult disease, because one of the things that I think about all the time is we have all these great new therapies for cystic fibrosis that are absolutely wonderful to the pediatric population. But I don't know how many of you guys have taken care of our adult CF patients. They're lovely, and they've already got lung disease. We're not going to fix that by potentiating CFTR today. We have to have regenerative therapies to try to improve that situation for them. And so targeting strategies like this with the gene therapy, it's not enough just to fix the cells. You also have to be able to activate them in the way that's going to generate a regenerative response. Okay, so last but not least, how do you actually make these things into functional clinical trials? I told you earlier that a number of clinical trials in critical care with new therapeutics have failed over the prior 10 to 15 years, to the point that many pharmaceutical companies have now just basically given up on doing critical care trials for new therapeutics. And the reason for that, frankly, is because we don't understand our diseases well enough, in my view. You need to, and among the things that we don't know are which are the right cells to target. We talked about that. Which are the right pathways to target. We talked about that. But we also need to know when's the right time to give a therapy. So a great example of this is I told you that these human progenitor cells respond really nicely to FGF7 and FGF10, or KGF and KGF2. Well, it turns out there's a therapeutic for KGF. And you can give it intravenously. It's delivered nicely to the injured lung. And you would think that that would be an awesome pro-regenerative therapy. The trial's been done. They gave KGF therapy to people on day 2 of ARDS. Does anybody want to predict whether that therapy worked or not? It killed people. So why did a good therapy that we know from good basic science that should work, that we can deliver relatively well, not work? Because we gave it at the wrong time. So you know what's easy to do? Enroll patients on day 2 of ARDS, because you've defined them. They have ARDS, you know they have ARDS, they meet your inclusion criteria, and you have some time to get them into the trial. Those things matter. It's hard to recruit patients for clinical trials. You have to make sure you have the right patient population and a relatively clean patient population. But then you take a pro-regenerative therapy and you throw it into the teeth of a massive immune response, and you wonder why the cells die and the patients don't do well. It's because you did it at the wrong time. So maybe you need to enroll the patients on day 2, but not give the therapy until day 7 or 10. Maybe you need to identify a subset of the patients that you want to enroll based on their clinical trajectory in the first half of the trial before you give them the therapeutic. These are all the sort of things that we actually have to understand to be able to do these trials correctly. And most of the stuff that's clonal, that's from diseases that are focused on a single cell that's given rise to a number of bad cells that are doing bad stuff, the oncologists are all over and are knocking out every single cell pathway with targeted antibodies to try to figure out how to fix that. To the point that now they're like fifth-line therapies for these cancers that used to be lethal 10 years ago because they've developed a number of different defense mechanisms, and we have new drugs against those too. It's amazing. In critical care, we're not going to be able to get away with that. We're going to have to be rational about the way we design these trials, which means we actually need to understand when the right time to give these things is, which means we need more data. The only people who can possibly understand that problem are physician scientists, because my scientific colleagues have no clue about the details of the clinical problems that we face on a day-to-day basis. The problem with clinical medicine is not that it's hard, but that it's really complicated, and there's a lot of nuance, and there's a lot to know, and experience benefits you a lot. And so we need to actually take advantage of that experience. That doesn't necessarily need to mean that you all have to run labs to be able to participate in this process. But what it does mean is that physicians need to be involved in research with physician scientists and scientists to be able to actually get any therapies. Because anything that was easy enough for one lab or one person to solve is probably already solved or on the way to being solved. And so what we need to do really is take advantage of our clinical acumen to be able to figure these things out. The other thing we have to do is we have to be sort of skeptical about stuff that seems too good to be true. And the reason I say that is that there have been a number of trials in critical care and elsewhere of cellular therapies where you squirt mesenchymal stem cells into a person and you hope that good things will happen afterwards. And unfortunately, much of the basic science in the cardiology field around these mesenchymal stem cells has now been retracted because it was a major scientific fraud. And there are a number of things that seem too good to be true in therapeutic regeneration and probably are. I find it hard to imagine that you're going to be able to take somebody else's bone marrow-derived mesenchymal stem cells, pull them off the shelf from a freezer, stick them in me when I'm critically ill, and that's going to make me better. It's possible, but if it did work, I'd rather have the drug of the good thing that those cells made, which could be controlled pharmaceutically and developed in the same way that our other therapies are, than somebody else's stem cells getting injected into me, because I think the risk-benefit profile of that's a lot better. And that's the other thing that physicians can really participate in. There's a lot of things that I'd like to publish that I think are cool that are way not ready to go into our patients, right? And so the difference between this is an interesting scientific finding and I'm ready to stick this in my critically ill ICU patient is pretty large. And so I think we have an important role in trying to bridge that gap between, oh, interesting, I'm ready to do this. Maybe it even went into a really big, important journal versus, okay, I'm going to give it to my sick patient, right? And probably my young sick patient, the one I really want to save, because they've got a 5-year-old at home, and I'd really like them to come off of ECMO, so I'm going to stick this thing in them. There's a huge amount of space between what's interesting scientifically and what's ready to go for clinical prime time, and we're the ones who have to be responsible for that difference, because ultimately we're the ones who are choosing to enroll patients in these trials or give these therapies. And I think it's really important to understand the scientific aspect of that. If we do all of those things well, I suspect we can actually design trials that matter and that might actually help people. And not everything we try is going to work, but I think that you could be nihilistic about it or you can try to do better. And our hope, I think, in my group, and I hope that I've convinced some of you to feel this way too, is that we should be trying to do these things not because they're easy, but actually because they're hard and because we're the ones who actually have the acumen to be able to figure it out. So just to leave you, I'll return to the key message I wanted to leave you scientifically, which is that there is a Wnt-responsive progenitor cell in the lung epithelium that drives a lot of regenerative biology, and that through a series of complicated mouse experiments, we were able to identify a human cognate of this cell, which we can now study and try to understand for scientific purposes, and that I hope will give us the opportunity to translate some of these findings. And then before I take questions, I'll just thank some of the folks who have participated in this work. I've had tremendous mentorship. Ed Morrissey was my postdoctoral mentor. Jeff Whitsitt is my K mentor at Cincinnati Children's, and Frank McCormick is my clinical and scientific mentor at the University of Cincinnati. And I've been very fortunate. This is my team at Cincinnati Children's. I have a resident and 2 fellows in my lab now, as well as a grad student and a senior technician who have all been great. And I want to acknowledge Dave Frank and Jared Dept. Dave is a pediatric cardiologist who has participated with me closely in the Wnt responsive epithelial cells. And Jared is a Ph.D. scientist who worked closely with Dave and I defining the niche for these cells. And with that, I'll take whatever questions you guys have. Thanks very much. Yeah, so I'll restate the question just so everybody could hear it. The question is what the relationship between these alveolar progenitors is and lung cancer. So we are actually doing a project right now to activate oncogenes in these cells. I would predict to you that a progenitor cell is a good candidate for a cell of origin for malignancy. Type 2 cells have been demonstrated to be a cell of origin for adenocarcinoma of the lung. It turns out that if you look in an adenocarcinoma of the lung, there's actually a massive amount of cellular heterogeneity among the epithelium in those cells. And so it's a little hard to figure out what's the chicken and what's the egg in those cases. I think most people in our field think that alveolar cells are the cell of origin for those adenocarcinomas, but the data for that is relatively limited. If you're interested in learning more about that, there's a lab in Baltimore, Tyler Jacks's group, that has done a tremendous amount of work. Wnt signaling is key to developing adenocarcinoma in mouse lung. And so the prediction would be that these cells will be really important in that. But I'll see the data in a year whenever my student finishes up. Okay, that's an awesome question. So just to restate for the people who are remote, the question was about what happens to stem cells when you passage them and whether passage cells that can be sort of grown up for clinical uses will be the same as the cells that you're studying in situ. I think that we know for sure that culture of any cell changes its phenotype. So one of the things that I didn't talk too much about is that when you manipulate, when you ask a progenitor cell to do what we're doing, when you put it into an organoid, it's actually a really tough day for that progenitor cell, right? You digest it up out of its native environment, you pull it out away from all of its friends, you send it over a high-speed fax machine, and then you put it back into a new environment and say, do stem cell things, right? That's actually really tough for a cell to do. It's a really high bar. If you just take regular lung fibroblasts and you stick them on plastic, 2 passages later, they have a totally different cell phenotype than they did before. So I don't know that anybody's done prospective identification of the precise changes that happen with mesenchymal stem cells specifically. That data might be out there. I haven't seen it. But my prediction would be, based on all the other data we have, is that there's going to be phenotypic drift. The other thing that's probably true is that the commercial products that are available are pools from a number of different patients that are combined together. And the good news about the pool is that if somebody in that pool is good, then maybe all the samples are good. But the bad news about that pool is if somebody in that pool is pre-malignant, then all of them are pre-malignant. And that's the thing that worries me, is not so much that we know what the risk of those cellular therapies are, but that we don't know. And that before we're ready to start giving them to our patients, we should know a little bit more. And I hope cell therapy works, because if somebody can prove to me that it works, I'll give it to everybody with ARDS, because we got nothing. But my concern is that we don't understand the system well enough to be able to sort out the details.
Thank you very much.
Thanks, everybody.
Bill, about 12 years ago, one of the former chairs started this tradition, and I've always wanted to swing this back. So for all of our outside speakers across the country, we want everybody to have a Louisville bat, and it has your name on there and the date.
Thank you so much. Thank you so much for the invitation. I really appreciate it. Thank you. Thank you.
We're going to take some pictures with Jason here, and
Yeah, Jason's here. And are you flying or are you?
I'm driving in my car's
Okay.
in the parking lot, so
So
I'm
you
not
can
in the
take the
configuration.
bat.
Yes, I can take the bat directly back.
Otherwise, we would mail it.
No,
So we don't get you in trouble.
Oh, yeah, the
That sort of
TSA
thing.
might not like the baseball bat.
They generally don't. Yeah, you're right. They generally don't.
Oh, perfect. I have this thing for you, so if you, wonderful.
Do you want to take a picture? Yes. Okay. Yeah, we need that.
Oh, yes, I need the bat.
Aren't you sure everyone in his lab has a blast? Here, we'll